about the insight regarding my talk, which I just gave in Florence, I think one of the major challenges with the use of natural killer cells or simply NK cells for immunotherapy has been the short half-life of conventional NK cells. So traditionally, if you were to take NK cells, they last around 10, 12 days. Does they have natural half-life in the blood? So if you were to adoptively transfer them into patients, that's how long they're going to live. And that's been a very, very big challenge in the field. If you are going to use a cell type which lasts only 10, 12 days, how do you expect that cell type to lead to sustained anti-tumor responses? One approach has been using multiple doses of the NK cell infusions, but the problem with that approach is that you often end up needing lymphodepletion before each infusion, but then also that creates a challenge in terms of use of lymphodepletion, which may not be applicable in many patients, use of multiple doses. So several years back, when I was a postdoctoral fellow in Dr. Todd Feniger's lab at Washington University, my work focused on can we generate, first of all, can we discover, and then can we on purpose generate these memory-like NK cells in humans. And we found that if you were to take conventional NK cells from peripheral blood of anyone, or including patients, and expose them to a specific cytokine cocktail, which includes IL-12, IL-15, and IL-18, they differentiate over a period of several days into what we call memory-like NK cells, also called cytokine-induced memory-like NK cells, SIML NK cells. So what's cool about these memory-like NK cells is that they can live for months. In a syngeneic mouse model, around 60, 90 days. And in patients, we have some data that they can live up to as long as 8 months. Typically, again, 60 to 90 days, but in some patients, we found that these cells can live a really long time, which is exciting and makes these cells attractive for adoptive immunotherapy, as I mentioned. They help overcome this fact that they can live that long, helps overcome one of the major challenges with the use of conventional NK cell, which is their short half-life. In addition to that fact, these memory-like NK cells also have better anti-tumor responses. They produce more interferon gamma when they are exposed or co-cultured with tumor cells. And then also they are better metabolically fit. They tend to migrate better into the tumor microenvironment, and they resist suppression by TGF-beta better than conventional NK cells. So all of these properties make them really attractive for generating CARs, or for that matter, even TCR knocked-in NK cell-based products, or any other genetic manipulation, if you like, for adaptive immunotherapy. So my lab at Dana-Farber for the last several years has been really focused on generating CARs and TCR-mimetic CARs using memory-like NK cells. And during this talk, I mentioned one CAR against NPM1. So NPM1 is the most commonly mutated gene in AML. And as you know, that in acute myeloid leukemia, or AML, there aren't any good targets. So most of the CARs which have been attempted against AML so far have mostly included CD123, CD33, and things like that. And they are also expressed by a normal hematopoietic stem cells. So there's a risk of myeloablation. So my group, in collaboration with a group at MIT led by Professor Jianru Chen, we identified a specific clone of an antibody which binds to the new epitope derived from mutant NPM1. And we call it peptide-centric, also called TCR-mimetic, or simply TCR-like CAR, because it kind of acts as a TCR because it binds to the new epitope expressed by HLA class A2. A2 is the most common HLA-A allele in Caucasians, around 55% of the population. So we successfully generated a CAR which worked very well, both in vitro as well in mouse models. So we are hoping to take that CAR to clinic because, again, AML has been so far very hard to treat with a CAR, and this specific CAR seems to mitigate the possibility of myeloablation, which is a major advantage. And then I mentioned the talk that, yes, in a mouse model, we can cure a lot of tumors, but many times in humans, we still fail. And how can we make CAR NK cells more effective, if you will, in terms of both enhancing their anti-tumor responses and making them live longer and expand better? So my group has been working on the use of IL-12 incorporating into our CARs. Now, IL-12 is not a new cytokine by any means in terms of immunotherapy. If you look back, the first trial was done, I believe, in 2002 or so, using human recombinant IL-2 systemically in patients. And there were some toxicities and things like that. And then later on, there were groups which actually incorporated IL-12 into CAR T cells, and those were associated with really, really bad toxicities, leading to early termination of the trials, and also led to a few deaths in those patients with severe CRS. So IL-12, despite the fact that it's one of the most activating, most powerful cytokines, has been associated with toxicity, and in general, in the T cell world, people are now shying away from using IL-12 in their products. We, in my lab, we actually compared IL-12 knocked-in CAR T cells versus IL-12 knocked-in. When I say knocked-in, I mean overexpressing secretory version of IL-12 CAR NK cells head-to-head. And upon tumor at baseline and upon tumor co-culture. And what we found was that the CAR NK cells, even when they have IL-12 incorporated, they produce way less quantities of key cytokines which are associated with cytokine release syndrome, including IL-2, IL-6, GM-CSF, and things like that, suggesting that CAR NK cells, which are well known to have better safety profile at baseline than CAR T cells, but even when you were to knock in a very powerful cytokine like IL-12, they still produce way less quantities of pro-inflammatory cytokines like IL-2 and IL-6, giving us some hope that IL-12 incorporation will be safer in the context of CAR NK cells. And then when we also, there are groups which have been using IL-15, for example, for enhancing CAR NK cells. So in the study, we actually also compare IL-12 secreting CAR NK cells versus the IL-15 secreting CAR NK cells, and we found that even though their short-term anti-tumor responses were similar, however, what IL-12 did better was it produced way more interferon gamma, more TNF alpha, more granzyme B, and better fast ligand expression. All of those things are important for meeting anti-tumor responses. But then also, we found that they get less exhausted upon tumor rechallenge compared to the IL-15 secreting CAR NK cells. Again, suggesting that IL-12 is a better cytokine for to enhance anti-tumor response called the CAR NK cells. And then also in the talk, I mentioned use of another target called mesothelin in AML. Now, mesothelin has been heavily investigated in solid tumor settings, including pancreatic cancer and ovarian cancer. And there have been trials both using immunotoxins as well as CAR T cells against those targets in those malignancies. However, it's less known in the hematological malignancies world. Work led by Professor Sahel Minchinchi group at Fred Hutch has shown that certain types of or certain subtypes of AML, including pediatric AML, as well as AML with KMT2A rearrangement, also called MLL translocated AML, as well as core binding factor AML, they tend to have aberrant expression of mesothelin. And because of that phenomenon, we generated a CAR which incorporates secretory IL-12 to make them, robust in terms of anti-tumor function. And we use mesothelin as a target, and we found that this CAR really works well both in vitro and mouse model, despite the fact we use very low dose of and single infusion, also low dose of IL-12 secreting CAR. And then last part of the talk, I mentioned use of CD70 as a target. Again, CD70 is a well-known target in AML and other malignancies, including some lymphomas as well as kidney cancer. And my group has ongoing studies in kidney cancer. But then in this particular talk, I mentioned how when we use the same IL-12 secreting CAR against CD70, it works really well. You must hope that that could be the other potential way to make progress in AML. And in summary, my group is very interested in AML and developing CAR and NK cells, particularly using the platform of this innate, memory, or also called memory-like NK cells, arming them with CARs, especially CARs which mitigate the risk of causing myeloablation. So going after targets like NPM1, which again is not shared by the normal hematopoietic stem cells, mesothelin, and CD70, all of them have this one common thing, which is none of those antigens are expressed by normal hematopoietic stem cells. So there's less risk of causing myeloablation as using CAR NK cell platform for better safety profile and incorporating IL-12 to make them really powerful. That was the talk about. I'm very excited about this work. Not only we are learning a lot scientifically, but also we are taking these products to clinic, and I'm excited to see that happen. And time will tell how they will perform in patients.